Hello. Last time we talked with you about neuroplasticity, about what it is, about where and how it works, and what are the major rules for neuroplasticity, most efficient development. Today, I'm proposing to talk about another interesting subject, which is how our neuroplasticity changes across the lifespan. From the very beginning of our journey on this planet and to the very end of it. So shall we start? First of all, we cannot stress enough that brain is a very complex organ. It is most complex organ in our body. And I will supply that with some facts. Our brain contains about 100 billion neurons. And each of these neurons can create a synaptic connection with 5 to 20,000 other neurons. You understand how big numbers we are talking? We are talking factorials of these numbers, including that one, the 100 billion. My calculator and my iPhone refuse to calculate this big of a number. And another fact, about 14,000 of genes is expressed in our nervous system, while there is only 20,000 genes being expressed in our body during the rollout. You see, it's a big share. Only 6,000 of this 14K genes is being rolled out, is being expressed only in your nervous system, and 8,000 in both nervous system and other tissues of our body. It's also important to mention that major forces which are driving our early brain development are these. You see them on these slides. So, force number one is genetics. Genetics is something that we cannot change, unfortunately, at this stage of our science development, but it impacts a lot the process of our brain and the entire body actually roll out. But there are two more factors which impact as our early brain development and also neuroplasticity throughout the lifespan. One of them is our sensory motor experience. It's an experience which we are gaining with our senses from the surrounding world. And another factor is a dynamical system. What does it mean? It means that our body is a dynamical system, and how this dynamics will go depends not only on genetics, it depends on a lot of factors. And major factors here are how we maintain our body. Do we eat the right diet? Do we exercise our body? Do we have any bad habits or no? Does our body get enough sleep, enough rest? All these factors are having a great impact on how the dynamics of the body works. Also, of course, our possibility to get medical help and you name others. Before we go to the next slide, I would like to mention that the rollout of our body and the brain is a self-organizing system. And here we have our first picture, which tells us about changes in the brain across the lifespan. Here on the left, you see the photo from a microscope, which shows the density of synapses at different phases of our life. So first one is at birth, and you see that density of synapses at birth is quite low. If you look at the graph on the right, it also shows the density of synapses throughout the lifespan, and at birth, at this point to be particular, it's quite low. If you're wondering what this red line is and what the black line is, so these are both graphs that are showing the density of synapses throughout life, but black shows the total density, and red one shows the visual area density. But basically they have the same manner of growth and fall. So then, after birth, we see that the number of synapses starts growing dramatically and reaches a peak at about 1 year old. The microscope picture shows us the 2-year-old's tissue of the brain, of the probably cortical area of the brain. And the density of synapses, as you see, is quite high. And then, after a certain point, it starts to fall down. And already, when the child reaches 6 years old, it gets twice less. You can see the corresponding picture from microscope on the left. By 10 years old, the density of synapses reaches to its state, to the plateau, which will last until about 30. And then it starts to decline till the end of the life. Of course, this decline is different for every person, and it very much depends on how this person lives his or her life, how he or she maintains the body, how he or she pushes her brain, his or her brain. And if their brain meets any new challenges, if they are exercising enough, we will learn more about what neuroplasticity depends on in later presentations and discuss what can influence the health of the brain, the development of the brain. And here we came to another picture, which shows mammal's brain. Here is the monkey's brain, which is very similar to ours. They also have neocortex, as you know. And graphs are showing different cortical areas and density of synapses in these cortical areas throughout the life of a monkey. So we can see the prefrontal cortex area, we can see the motor cortex, somatosensory area, visual cortex, and limbic system. And as you see, the density of synapses behaves more or less in a similar manner. The prefrontal cortex gets to its high also at a certain age, and then it plateaus more till the rest of the life, while the visual cortex peaks and then declines with greater degree. But what I like about all this picture is that philosophical sense that it has, metaphorical sense that it has, that it carries. If you look at all those graphs, at once, and you see that you can think of our brain as a kind of computer system which gets rolled out throughout life. Well, if you like it as much as I am, you can return here later and just have a look at it and maybe come up with some reflections, which you can put down in the comments. And we move on to the other slide. So as long as we are talking about brain development across the lifespan, we should mention the critical period. It's a period of our nervous system development during which the nervous system is particularly sensitive to the effects of experience. So if you think of it, the normal sensory motor experiences that we get influences formation and maturing of neuronal networks, while the failure to be exposed to an appropriate stimuli during the critical period might be irreversible or hard to remedy subsequently. We will look at the examples of those. Sensory motor anomalies during early critical periods can cause subsequent functional impairments, and we will look at examples of those too, such as an autism development. So if we talk about the most common example of how the failure to be exposed to a certain stimuli impacts further development of our nervous system, we can talk about a very common experiment which is being done to kittens. Usually, the researcher is closing one of the eyes of a kitten for some months in the early life of this kitten. And then, after usually 3 months, when they open this eye, and although this eye is physically perfectly healthy and theoretically can be a good instrument to get visual senses, visual information and process it, still, since this eye was closed during the cortical maps formation for a kitten, the cortical maps were visual cortex maps were already formed with just one eye. So this eye that was closed will never be a proper instrument for this kitten to get the visual information. Let's move to another example. And this is a learning language experience. We have a graph on the left which tells us about relative fluency, which depends on the age. And we see that relative fluency on the level of native speakers can be reached only if child was exposed to a language in his or her early years. And that would be until 3 to 7, depending on each individual case. Later on, the capability of a child to learn language at a native speaker's level starts to decline. And as you see on the right picture, which is a fMRI picture, you see that cortical areas which are activated during language learning, during words recalling, is quite different for children at age 7 to 10 and for adults. You see that when the adult is trying to recall foreign language word, the cortical areas which are being affected by that are much vaster than the cortical areas which a child is using. Here you see. So whenever the child is being exposed to a language in the earlier age, we can conclude that they use their cortical areas for recalling the words of this language more efficiently. Now let's look how early brain anomalies can affect the symptoms later on in life. Here we have a graph which shows the brain size for people with autism spectrum disorder. And you see it's this lilac color, dotted line. And the solid line in beige color is a typical brain development. And we can conclude here that brain overgrowth, and usually we speak cortical areas here, frontal cortical areas, overgrowth in early age causes symptoms which we call autism spectrum disorder. Another example is brain development of kids with ADHD. So we all know about ADHD. We call it hyperactivity in short, but in full, the abbreviation means attention deficit and hyperactivity disorder, which is quite happening with kids. So the fMRI diagram on the left shows us the cortical areas which are being affected for kids with ADHD. And on the right, you see the graph of cortical thickness. In red, we see the cortical thickness for normal brain development, normal cortical thickness. And for kids with ADHD, this cortical thickness is less than normal. Therefore, we have another example how the brain anomaly can impact the symptoms which a person can develop. And now, let's talk about how our brain changes across the lifespan. Before we do that, let's understand what we mean when we say white matter in the context of the brain, and when we say gray matter in the context of the brain. So when we say white matter, we usually mean axons and myelin, which covers axons of our neurons to protect them. And when we say gray matter, we usually mean neuronal bodies, dendrites, and glia cells, which are also are constraints of the brain. And here to the left is the picture, which shows us how gray matter develops over the lifespan. So in the beginning, the warmer the color, the higher the density of gray matter is. And you see that in early age, at the age of 5, there are much more gray matter areas in the brain. And then the brain starts to get myelinated. And axons, the connections, the synapses are becoming more stable. And by the age of 20, this myelination completes. And we consider that brain is more or less formed, and the gray matter volume gets less, while white matter volume gets higher. And here is a very interesting graph which shows the human brain throughout lifespan. We have distinction between female brains in red and male brains in black here. So they start more or less the same, and then they start to get a little bit different at the age of 2. So male brain weight is a bit higher than female brain weight. And we see that by the age of 20, the brain weight gets to a plateau, which lasts till the age of 50 approximately. And then it starts to decline. It's worth mentioning that currently it is considered that normal development of a human brain does not assume loss of neurons. So this normal decline happens because of loss of myelin, first of all, and loss of water, that's second. Here you can see also a couple of graphs on the right, which show how the white matter and gray matter developed across the lifespan. As you see, gray matter declines over the age, first pumps up a little and then declines in both males and females. And white matter, on the contrary, it grows over the age. So white matter, as we remember, is axons and myelin. So it means that by the age of 20, our brain becomes more stable, less flexible. And finally, one of the hot subjects about the brain development across lifespan is neurogenesis. We hear a lot of talks about that. Some people say that neurogenesis takes place in adults' brains. Some say that it doesn't. So let's find the truth about what science knows for the moment about neurogenesis in adult brains. So what we know for the moment about neurogenesis, that yes, it has its place in the adult brain, but it only happens in a couple of areas of the brain. It happens in olfactory bulb and the hippocampus. Hippocampus and olfactory bulb are the only sites of adult neurogenesis. In both of these brain regions, new neurons are generated by neural stem cells, which are retained in restricted locations of our brain. Some of these neurons become successfully integrated into functional circuits of our brain. However, unfortunately, most of new neurons die before being integrated into existing neural networks. No one yet was able to explain what is the functional significance of this restriction. Why new neurons are only being born in the 2 areas. And also, there is another restriction for these new neurons. these new neurons are only GABAergic, and they are only interneuron. This limitation hasn't been explained by now either. So what are the perspectives of the neurogenesis in our brains? Preliminary research results show already now that neurogenesis in hippocampus of an adult person can be modulated by, first, medication. Well, for that, we have to take advice from our doctor. And the second one is by physical exercises. So this is something that we can deploy. Any one of us can deploy. And it is strongly recommended for the sake of our brain health to exercise on a regular basis. The challenge of developing this capacity to generate new neurons and circuits is very interesting for a lot of researchers around the world. And it can help to repair after brain injuries or be a remedy to degenerative diseases. So I guess that scientists will keep working at these areas and find out the new ways to boost neurogenesis in adult brains. And I thank you all for your attention today and suggest that we all go now and deploy some physical exercises which will help us generate new neurons in our hippocampus, which hopefully will be deployed in some of the existing neural networks. Thank you.